This is Dante, Dehnoushi, and James Weller, and this is Disinfectants versus Bacteria.
Our question was, how well will disinfectants fight bacteria? And our hypothesis was that the disinfectant with the highest alcohol percentage will clean the best.
We are testing different disinfectant solutions and products to find out which one is the best for different purposes and which one will truly disinfect the most bacteria. Bacteria are single-celled or simple organisms that are invisible to the naked eye. Many bacteria are found both inside and outside of organisms, including humans. Bacteria are also found on surfaces and in substances like water, soil, and food, making them key players in the Earth's ecosystems. We are looking for the best and most effective bacteria cleanser. Our experiment will ensure that people are buying the right products for their everyday cleaning use. This will create a cleaner and safer environment for people around the world to live in. We did this by using LB broth to help the bacteria grow. Bacteria feed on LB broth, which will cause it to grow over time in a fast manner. After letting the bacteria grow for a few days, we added disinfectants to the bacteria using a swab to see which disinfectant would clean the most bacteria. We thought that the parasitic acid would clean more bacteria than the Windex and chlorine. That is because of the amount of alcohol in the parasitic acid. Windex has 4% alcohol and chlorine has 8% of alcohol, while parasitic acid has 15% of alcohol in it.
Our materials were 9 petri dishes, Windex, chlorine, parasitic acid, bacteria, and swabs. And our procedure was that first we took the LB broth and poured it into 9 different petri dishes, 3 per disinfectant. And then we rubbed the bacteria on the LB broth so that the bacteria could feed off the LB broth. And then third, we waited 4 days for the bacteria to grow. And then fourth, we added some of the disinfectants to the petri dishes with the swabs. And then finally, we waited to see how much the disinfectant would affect the bacteria.
Our hypothesis was supported by our data in which the disinfectant with the highest alcohol content had to clean the most bacteria. The plates with acid added into them cleaned out the most bacteria. Alcohol kills germs through a simple chemical process known as denaturation. Denaturation occurs when alcohol molecules break down the proteins present in the structure of germs. When the proteins break down and lose their structure, the cells can't function properly. They lose their membrane protection, dehydrate, and quickly die. We measured our bacteria by counting the amount of bacteria colonies in each Petri plate. The average amount of bacteria cleaned from the Windex group was 26.2412%, while the average of parasitic acid was 42.8311%, and the average of chlorine was 21.346%. When we first tried this experiment, our lab failed and our bacteria did not grow, but because of Mr. Heinen's amazing generosity, he let us regrow our bacteria and collect the data again. While collecting our data, after our failed first attempt, we realized that bacteria grows in many varying ways. Depending on what you do, different types of bacteria will grow. They also vary in how fast and how much they grow over a period of time. Some will grow fast and grow little, while others will grow slow and grow a lot of bacteria.
In our experiment, we wondered what disinfectants would work the best against bacteria. In the end, we learned that disinfectants with the highest amount of alcohol did the best at breaking down bacteria. We learned many things while conducting this experiment. We learned about how bacteria works and how it grows and gets broken down. After failing on our first attempt, we learned that the reason it failed was because of the conditions we left the petri dishes in. During the night, it was really cold in its environment, so the bacteria wouldn't grow. While cold temperatures don't necessarily kill the bacteria, they can slow or stop the growth of it. This means the bacteria won't reproduce quickly, but it also means it won't be completely destroyed. To prevent this from happening the second time, we left the petri dishes in a cardboard box during the night. If we were to do this experiment again, there are a few things we would change. We would leave the bacteria to grow for a longer period of time before adding the disinfectants. We came to the conclusion that our experiment was inaccurate because the results were all over the place. Even though we did 3 tests for each group, our results for each group were very different. Next time, we would like to try and find a way to make it more accurate so we can find the best answer correctly. We would also like to try out different types of disinfectants. Overall, this experiment was a lot of fun, and Dante and I had an amazing time learning about how bacteria and how it grows.
5.
The top data table shows the average percentage of change between the 3 disinfectants, and the bottom data table shows the amount of colonies before and after between the 3 disinfectants.
The graph on the left shows the average percentage of change of the bacteria, and the paragraph on the right shows the colonies before and colonies after we put the disinfectant in.